I would like to immediately continue to introduce Shiva Shivashankar from the Paul Scherrer Institute, and he has a joint appointment with us at ETH in Health Sciences and Technology. The floor is yours, Shiva.
Great. So let me thank the organizers, in particular Viola, for having me here. It's a great conference. So what I'm going to talk to you today is about thinking about how to connect the extracellular matrix that Viola talked about, and how does it feed onto the genomes, and how do genomes respond to this. As a disclaimer, we recently co-founded a company. It's an ETH spin-off, and what I'm going to talk today is some aspects of it is also part of the company. So as all of us know, cells differentiate into different cell types in the body, from stem cells to various different types of cells. And one of the key elements in this process is the same DNA, but it's the same DNA that then packs in different nanoscale architecture, and that nanoscale architecture then gives us all kinds of cells which adapt to different tissue stiffness. And so all of this actually changes as we age, because aging is inevitable. As cells age, as organisms age, this organization, the nanoscale organization, the genome structure changes. And this leads to a number of aging-related diseases. Around the world, there's been a number of efforts now to try and revert aging. One of the major efforts is through the induced reprogramming that won a Nobel Prize for Yamanaka and Gurdon. And there are variants of that, either using biophysical signals with the Yamanaka factors or chemical molecules. But however, in our tissues, cells age, and you don't have these exogenous factors. So what that means is that the local tissue microenvironment has a lot of information, as Viola pointed out, and we must learn that information if we want to revert aging processes. So that's what I'm going to talk to you today. And as cells age, the genomic organization, the nanoscale organization, changes dramatically. And somehow, if we can reset these two processes through the extracellular matrix constraints, maybe we have a chance to really think about regeneration processes. So in that context, I'm not going to belabor this point that Viola did, that cells within our tissue really recognize all these important extracellular microenvironmental signals, but that gets relayed down to the intracellular organization, as well as the genomic architecture. So there's a very tight coupling between what cells interpret outside and what cells interpret from the genomic programs to be able to provide an appropriate feedback for cells to maintain homeostasis. So can we use this information to try and see if we can revert aging processes? So one of the things that we figured out was that cells, if you understand what they see outside, and if you manipulate it by various engineering methods of how the cells can actually sense the extracellular environment, what we realized was that we could take a somatic cell, engineer its microenvironment such that you push these cells into proliferation. And as the cells proliferate, you have a very default de-differentiation pathway. This seems to be a very generic principle that we have discovered, that cells, if you impose the right microenvironmental constraints, they have a de-differentiation pathway. And so we could show that these cells really go into this de-differentiation and eventually, after a few days, start becoming stem-like cells. So you don't have any exogenous factors nor chemical factors. All you've done is identified an appropriate constraint for a cell so that these cells can actually start dividing more, start losing the memory of the somatic state. So this is exciting to us because if you now take an aged cell, as many of you know, as cells age, cells accumulate a lot of defects. So reverting aging cells means that you need to clear all these defects to a healthy state. So one idea was that if you are able to de-differentiate a cell, do you lose memory of this aging effects so that then you can actually have a cell where if you re-differentiate it back, you have a healthy cell state or a youthful cell state. So that's the hypothesis we went after, and we've had a lot of luck with this. The main idea is that, to cut a long story short, if you re-differentiate these partially reprogrammed cells, what you find is that you have a gain of function. As these cells re-differentiate back, what you start finding is that they gain all the youthful cell states. This is exciting to us because what you have done is you have taken an aged cell, figured out what the microenvironmental constraints that needs to be, and imposing such microenvironmental constraints, what you have made is a group of cells which are stem-like and re-differentiate them. And what you gain is a youthful cell state. So we're not trying to understand the mechanisms that underlie this because we feel that this is an important step going forward to understand cell state transitions. And some of the ideas that we developed over the years for this is in these publications. So the goal then is what you have is a possibility now to use this as cell-based therapies because you can actually derive patient-derived cells from an older patient. You partially reprogram it through these mechanical constraints and implant them back to the same patient. Because that's the biggest challenge in cell-personalized medicine is that you need to really implant back the same cells from the same patient to have actually matching of the immune processes. So we've done that now in thinking about skin tissue regeneration, and we've had a lot of luck there. These are engineered skin tissues. You can take this patient-derived dermal fibroblasts, partially reprogram them, and implant them into the tissue, and ask if these tissues heal better. Is the wound healing better? Because as we age, wound healing is compromised. And now we are trying to see if we can implant these kind of cells in different organs. We're very excited about it. Happy to talk offline if anybody is interested. So one of the challenges going forward for this is once you implant or once you age, if you really want to understand how cell state transitions are happening, these nanoscale changes that are happening, you need really good biomarkers, which is informing you of the cell states as they age. And if you want to revert it back, you need this appropriate biomarkers. So we looked at, because the nanoscale architecture of the genomes are so central to defining cell state, its health and disease, and as has been annotated for a number of years, that architecture changes in aging-related diseases and in most diseases. What you see here is changes in premature aging, in Alzheimer's, and in cancers. In all of it, one of the general things that happen is that because of the changes in the extracellular environment, that reflects in a robust change in the nuclear organization, in the genomic architecture. So we thought that if this is what is telling you about the cell state, then we should be able to combine this with a lot of machine learning techniques and learn those pictures of how our genomes are constructed in our cells, in intact tissues, in human tissues. So this has led us to a very nice program of biomarkers where we can actually take tissues or cells in culture and image them with simple microscopes and learn how the DNA or how the genomes are organized within our cells and tissues. Using this now, what we can construct is an elaborate program of the cell's health as we age, or as we age, how the cells transition to various disease states. And in addition to that, what you need is, if you want to connect it to function, you need to be able to go from simple images to a lot of functional information of the cells. Because there's a whole area of looking at every cell in a tissue and do proteomics, transcriptomics, and so on and so forth to understand how we age, how cells age in tissues, how different diseases arise in tissues. But this is a very daunting task. To be able to measure every cell's information in complex human tissue biopsies, tissue microenvironments, is a very complicated task. So what we have now come up is methods of assessing the nanoscale genome architecture that exists in those cells and convert them using a number of machine learning techniques to other types of data that exist for the cell. It could be transcriptomics, it could be proteomics, it could be regulatory networks. So this gives us now a handle on thinking about cell states in intact tissues, and we have now applied this into multiple contexts. One major context is, for example, as we age in neurodegeneration, we can now fingerprint in postmortem biopsies, both in mouse as well as humans, looking at the genomic architecture in intact cells, which regions are poised for protein aggregation in terms of the amyloid deposits. And in addition, we can now look at tissue biopsies of tumor progression. For example, this is a breast tumor progression. And in the tumor progression, you can now fingerprint every nucleus, its health, its transition from its normal state to a tumor state, and as the disease progresses. And more recently, we have now been able to look at blood cells using liquid biopsies, and look at the chromatin structure, the genomic structure in blood cells, and really assess, can we detect a tumor in the body by just looking at the blood cell chromatin? And if there's a therapy done for an advanced tumor, can we detect it directly using a simple blood cell chromatin structure? So this has also given us a very good insight on thinking about, because such signatures show up in the blood cell genomic architecture, this is a very scalable approach for thinking about human diseases. So we now can actually detect different types of cancers, healthy versus cancer patients, with very high accuracy, but just looking at the genomic architecture, that suggests that we should be able to scale up to a public health project. So what we're now initiating is a large public health initiative to be able to look at a blood cell chromatin structure and develop very simple microscopy techniques on a mobile phone so that you can look at this blood cell chromatin structure and assess patient-specific information about disease progression. So what I said so far, since my time is also running out, is that by thinking about a simple idea of how cells interact with the extracellular environment, what Viola described, where there's such a rich information of how cells sense that, and looking at how the DNA is organized in each of our cells, and looking at the changes in the DNA organization that comes about because there's mechanochemical information that gets transduced from the external environment into the genomes to be able to bring about differential expression programs. And I also told you that this idea of looking at the genomes and the regulatory network, combining with machine learning techniques, it gives us an exquisite fingerprint of every cell in our body. Right? So we're now scaling that up into a large-scale program. So I recently moved, not so recently, moved to Switzerland in 2020. The goal of our group is to try and understand how, in aging processes, how does this nanoscale architecture change? Can we revert these architectural changes so that we can have gain of function, regenerate aging cells, and use such architecture as biomarkers for cells' health and disease? So with that, I'm going to end. I've been very fortunate to have very talented colleagues coming from biology, engineering, and computer science, and a number of collaborators. Most importantly, this work, I'm also affiliated with the Broad Institute now, is carried out in collaboration with Caroline Uhler's group. Our groups work together With that, thank you so much.